Hello, I'm Licia Iacoviello. I'm the head of the Department of Epidemiology and Prevention of the IRCS Neuromed in Pozzilli, and I'm Professor of Hygiene and Public Health at the University of Insubria in Varese. The topic of my talk today is what we learned about coagulation from population studies. And I will try in the next 30 minutes to give you some evidence that not only it is possible to learn a lot on coagulation through epidemiological studies, but also I would like to give you some evidence that there is a sort of common soil that we can derive from such studies between cardiovascular disease and cancer. Normally, we consider these two diseases as two different trees, but they embed their roots in a common ground. So we can identify in hemostasis in general, in coagulation in particular, a common soil that is at the basis of the risk of these two diseases. This is a very simple scheme of the coagulation cascade and the following fibrinolysis activation is just to focus the attention on the different aspects of the cascade that have been interested by epidemiological studies. From its beginning, the exposure of tissue factor after a cell perturbation like endothelial cell or even monocyte or granulocyte circulating into the blood, this perturbation exposes tissue factor on the cell membrane surface, and tissue factor links factor 7, activated factor 7, and starts the coagulation cascade. All this process ends with the lysis of fibrinogen in fibrinogen monomer and then polymers, thanks to the activity of thrombin, that is the central enzyme of this cascade process. But in this complex process, the same enzyme that activates thrombosis, inducing the lysis of fibrinogen, also activate the counteracting process. So the fibrinolysis that is deputed to dissolve the polymers of fibrin through the action of plasmin that is generated by plasminogen through the activity of another enzyme, the tissue plasminogen activator, that is in turn inhibited by PI1. Once generated, plasmin is able to lysis the cross-linked fibrin, generating D-dimers. Again, D-dimers, once increased in the blood, is the expression of the activation of two counteractive systems, the coagulation from one end and the fibrinolysis from the other end. This is very important because we have always to consider when we speak of coagulation, and in general, when we speak about blood activation systems, that the systems is from one end activated and from the other end inhibited. Let's start from the end of the cascade. So the formation of the dimers, that, as I told you before, is the expression of both the activation of coagulation, so thrombin formation from one end, and activation of fibrinolysis, so plasmin formation from the other end. We study the levels of the dimers in circulating blood as a possible risk factors for cardiovascular disease in the frame of the Molisani studies. Just briefly, I explain you what is the Molisani study. It's a cohort study. They started in 2005 and recruited between 2005 and 2010 over 24,000 people from the general population. So they were apparently healthy people from the general population that were studied at baseline and then followed up for almost 10 years for a number of disease incidents. First of all, mortality for all cause and cardiovascular and cancer causes, but also incidence of cardiovascular disease and cancer. So this study could be used to simultaneously evaluate the association between an exposure, in this case factors belonging to the hemostasis or in particular to the coagulation system, and an outcome, mortality or cardiovascular disease or cancer altogether. As you can see from this graph, increasing levels of circulating D-dimers were associated with an increase in the risk of all-cause mortality. But the association was not linear. Indeed, only in the presence of D-dimers level over 232 nanogram ml, the increase in the risk of mortality was evident. But we have to take into consideration that we are speaking about a general population and normal population, so we are speaking about a normal distribution of the D-dimers levels and not on a disease condition. A validation of this association in another epidemiological context was given by the EPICOR study. EPICOR is the Italian cohort study of the larger EPIC study, where not only association with cancer disease, but also association with cardiovascular disease were studied. We had the possibility with a nested case cohort study to reveal that the dimer levels were also associated with the risk of stroke in a positive manner. So increased levels of the dimers were associated with increased risk of stroke, and in particular with all types of stroke, but also with ischemic and hemorrhagic stroke. Again, this relation proved that the activation of the coagulation system and the D-dimer production can be involved in the process of thrombosis, but also in the formation of hemorrhages. This because it is a mirror of the coagulation activation in the case of thrombosis and of fibrinolysis system in the case of hemorrhages. Not only the dimers levels are associated with the risk of ischemic arterial disease and hemorrhagic arterial disease, but they are also associated with the risk of cancer in women. In the slide, you can see as high levels of the dimer in the normal range of the distribution. We are speaking of level higher than 234 nanogram ml DL, were associated with an increased risk of breast cancer in age-adjusted analysis, but also in a multivariable model where age, metabolic syndrome, deep vein thrombosis, contraceptive use, and menopausal status were used as confounding. What is important when we discover an association between an exposure and the risk of a disease is have the possibility to demonstrate that the levels, the increased levels, and the situation can be changed by changing lifestyle habits. Again, in the framework of the Molisani study, we were able to demonstrate that people who adhere more to the Mediterranean diet, so have an healthy diet, also have a decrease in the high level of fibrinogen of the dimer. So people adhering more in the highest decile of adherence to the Mediterranean diet had a 50% probability to belong to the high level of the D-dimer distribution. From the scheme I presented you in the first slides, we derived that D-dimers are the products of the activity of thrombin on fibrinogen, and then the activity of plasmin on the fibrinogen dimers and polymers. In a collaboration with the Synapse Research Institute, we had the possibility to measure the fibrinogen level in the plasma samples of our population and to associate these levels with the risk of mortality. So it is already known that increased fibrinogen level are associated with the risk of cardiovascular disease. And we also know that fibrinogen is a molecule that is not only relevant in the coagulation system, but it's also relevant in the inflammation system. So increased levels of fibrinogen can be a proxy of activation of coagulation, but also a proxy of high level of inflammation. What we performed in our analysis was to compare the risk of all-cause and cardiovascular mortality with the normal level, using normal level of fibrinogen as the reference class. So giving the possibility to understand or to evaluate not only if high level of fibrinogen were associated with mortality, but also if low level of fibrinogen could be associated with mortality, using low level of fibrinogen as a proof of a subchronic activation of coagulation that slowly, through this process, consume the levels of fibrinogen. And indeed, as you can see in this slide, we confirm that high level of fibrinogen were associated with an increased risk of all-cause and cardiovascular mortality, but also we discovered a trend to an increased risk of total mortality for low level of fibrinogen. So investigating deeply this association using spline curve that can give us a picture of the dose-response relation between fibrinogen and mortality, we could make evident a new shape relationship between fibrinogen level and mortality. And this relation was much more evident when we separated women from men. Indeed, in our female population, not only high level of fibrinogen were associated with an increased risk of mortality, but also low levels of fibrinogen. We hypothesized that this low level of fibrinogen condition masked the presence of a subchronic activation of coagulation that consumed fibrinogen. So the high level became low level and increased the levels of the dimers that demonstrated the presence of an activation of the coagulation. And this was particularly evident in women because, as we already know, there is a strong relation between female hormones and the coagulation system. And in the presence of female hormones, and in particular estrogens, there could be an activation of the coagulation system and perhaps a subchronic activation of this system. But fibrinogen is not only associated with risk of cardiovascular disease, but it is also associated with the risk of cancer, and in particular of colorectal cancer. We could demonstrate that high level of fibrinogen, higher than 400 milligram DL, induced a 2 times increase in the risk of colorectal cancer. This was present both in men and women, but again, the association between fibrinogen and colorectal cancer was stronger in women. Going back to the starting point of the coagulation cascade, we have several evidence from epidemiological studies that also tissue factor is involved in the risk of cardiovascular disease like myocardial infarction or stroke. There are very few studies that evaluated the antigen circulating level of tissue factor. The EPICOR was one of these studies. And again, with the case cohort nested epidemiological design, was able to show that high level of circulating tissue factor were associated with a 2 times increase in the risk of stroke. And in particular, the association was evident for ischemic stroke, but not for hemorrhagic stroke. Again, showing that is the activation of the coagulation system leading to thrombus formation that is associated with stroke. The increase in tissue factor and the relation between tissue factor and stroke is genetically regulated. Indeed, in the presence of polymorphisms that are able to decrease the level of tissue factor, together with a German colleague, we were able to demonstrate that the genotype of a polymorphism in intron 5 of tissue factor gene was associated to a decrease in the risk of stroke in a combined Italian and German population. When we calculate the haplotype between the polymorphism in intron 5 and another polymorphism in the promoter of tissue factor gene, again, we were able to see that H3 haplotype was associated with a decrease in the risk of ischemic stroke in a multivariable model, so adjusting for confounding. And these polymorphisms were also associated with a decreased level of tissue factor. So less tissue factor is present in the circulation, and reduced the risk of stroke is present. To deeply investigate this relation and better understand the relation between inflammation from one end and activation of coagulation through tissue factor, we performed another study in subject with myocardial infarction or ischemic stroke at young age, where the effect of the genetic factor can be more evident. And we studied the polymorphism in the promoter of IL-1 beta gene, so a gene that codifies for an important inflammatory factor. People with the TT genotype of this polymorphism show the decreased risk of both myocardial infarction and stroke at young age. When we try to investigate the mechanism at the basis of this protection by studying lymphomonocyte cells derived from the blood of the patients we studied before, we could see that when these cells were stimulated with LPS, that is a strong stimulator of the inflammatory response in lymphomonocyte cells, we could see that the people with the TT genotype that was previously associated with a decreased risk of stroke or myocardial infarction was associated with a reduced release of IL-1 beta after LPS stimulation. But this reduced inflammation leads to also a reduced release of tissue, or exposure, sorry, of tissue factor on the surface of the lymphomonocytes, and then a reduction in the tissue factor activity. So finally, this cartoon depicts the relation between inflammation, coagulation, and risk of stroke. The perturbation of cells by a number of factors like LPS, but also oxidized lipids or shear stress, hypertension, or homocysteine, activate lymphomonocytes to produce and release IL-1 beta, a strong inflammatory mediator. IL-1 beta returns on lymphomonocytes or on endothelial cells and stimulated the exposure of tissue factor on the cells, so activating the coagulation cascade. However, simultaneously, IL-1 beta induce the synthesis of PAI-1, that is an inhibitor of the fibrinolytic system, so trying to upset the hemostatic system in the phases of coagulation and then thrombus formation. And PAI-1 is in turn an important risk factor common to cardiovascular disease and cancer. Again, in the EPICOR study, we measured the levels of PAI-1 in the circulation. Inducing the common design, we associated, using the same group of control, the levels of plasminogen with the risk of breast cancer, colon cancer, acute coronary syndrome, and ischemic stroke. Increased levels of PAI-1 were associated with an increased risk of all these diseases. So cancer from one end, ischemic coronary disease, and cerebral artery ischemic disease. And this in a model adjusted for a number of confounders, including biological factors. The activity of PAI-1 is the inhibition of tissue plasminogen activator. That is the main activator of plasminogen in plasmin. Also, increased levels of TPA are associated with the risk of hormone-dependent cancer like breast cancer. We can see from this table that levels of TPA in the fourth quartile of the normal distribution induce a twice increase in the risk of breast cancer in a minimal adjusted and in a fully adjusted model. And if you look at the continuous relation, we can say that each standard deviation increase in the TPA induce a 30% increase in the risk of breast cancer. What is important again in women is the relation between hemostatic factor, in this case TP1, and hormone estrogen. Indeed, the effect of TPA in inducing the risk of cancer is much stronger in women with breast cancer positive to estrogen receptors. But we can ask why fibrinolysis and coagulation should be involved in the process of cancer formation, growth, and metastatization. And one explanation for this question is that the main effector of coagulation and the fibrinolysis system, so fibrinogen and its polymers, are the substrate of cell growth from one end and on cell migration for metastatization for the other end. So all the factors that are in some way related with this scaffold of fibrin formation and dissolution are involved with the possibility of cancer cells to grow and to migrate into the tissue. I would like to conclude my talk giving some insight on the possibility to use epidemiological studies to study the association between thrombin generation, in vitro thrombin generation, and the risk of mortality of cardiovascular or cancer disease. The system of thrombin generation that has been studied by the collaboration with the Synapse Research Center on the samples of the Molisani study can give us an idea of the thrombin potential that is present in our blood. I don't want to spend too much word on thrombin generation that has been presented much better than me by other speakers in this course, but just I want to give you an idea of the graph that depicted the process of thrombin generation in whole blood. When you add the tissue factor, that is the activator of coagulation, to the blood sample, you at the end generate thrombin. And this graph depicted the velocity in which the thrombin is generated. And of course, higher is this velocity, higher is the generation of thrombin, and the total volume of the thrombin that is generated. So we can say that the higher is the thrombin generation, the higher should be the risk of thrombus formation. And on the contrary, the lowest is the thrombin generation, expressed as a prolongation in the time of thrombin formation or decrease in the area under the curve of thrombin formation, then lower should be the risk of thrombosis. And then we, in a cross-sectional design, try to analyze the risk factor for cardiovascular disease that could be associated with the endogenous thrombin potential. And indeed, we found that risk factors that were associated in previous studies with a decreased risk of cardiovascular disease of thrombosis were also associated with a decreased thrombin potential, like addition to the Mediterranean diet, higher education. On the contrary, risk factors that were associated with an increased risk of thrombosis were also associated with an increased thrombin potential, like the presence of lipid disorders like hypercholesterolemia, high level of BMI, high level of waist to hip ratio, the use of oral contraceptives, and so on. However, when we associated the parameters of thrombin generation with the risk of cardiovascular mortality, we had some apparently unexpected results. Indeed, our results show that a decreased thrombin potential was associated with an increased risk of cardiovascular mortality, while increased thrombin potential was not. And these results were also shown for another parameters that depicted the thrombin generation, so the lag time, in a study performed by the group of Tenkate in in Maastricht, where again a decreased thrombin potential as demonstrated by an increased lag time was associated with the increased risk of mortality. So apparently these results are quite strange, because we are saying that if we have a reduced thrombin potential, we have an increased risk of mortality, and in particular of cardiovascular mortality. But these results are only apparently in contradiction with the role of thrombin in thrombus formation. If you think what we have shown before, indeed, if we have a continuous activation of coagulation and then of thrombin generation in a subchronic, subclinical way, we induce a consumption of the factors that normally are active in the system. So what we expect? A reduction in the thrombin generation in vitro. Not because thrombin is not important for cardiovascular disease, but because thrombin has been already consumed in the chronic activation of the system. So I would like to conclude my talk with few messages that we can derive from epidemiological studies, and in particular from the Molisani study. First of all, coagulation and fibrinolysis activation are common pathway for cardiovascular disease and cancer. And this is very important because by decreasing coagulation and fibrinolysis through lifestyle improvement or drug therapy, we can prevent both cardiovascular disease and cancer. We can use markers of coagulation and fibrinolysis to improve the prediction model of both diseases. And what is important is that the activation of coagulation and fibrinolysis in a chronic way in vivo could translate in a reduction of coagulation factors. And then when we study the in epidemiological setting, the association between factors belonging to system and the risk of disease, we shall take into consideration the possibility to get these contradictory, apparently contradictory results. Thank you very much for your attention.